I know a lot of people, especially those that may have been around and living with the disease for a while, are always interested in what's happening in the research landscape. What are we learning? How are we applying what we're learning to new things? And it's really delightful, and it's my honor to welcome our two presenters today. But I'll start off with Dr. Choi, who joins us from Northwestern University in Chicago. And he's in and out, so he made special plans to join us today, which I'm just thrilled and delighted. So I'm not going to say too much here. I think Dr. Choi will really bring a lot of really interesting updates on the research to everyone and share what's happening behind the scenes, because it's pretty exciting stuff, I just got to say, right?
Yep.
So off you go.
Thank you very much. So I have to say is that it's just a tremendous honor and a pleasure to be here. I've dedicated my life basically to treating patients with disease and also trying to find new therapies for this disease. And it's always a wonderful experience to interact with patients and advocates. And this is such an important group that'll be critical in ways I'll describe for how the field has to move forward. So I just want to first start by just introducing myself. Some of my patients are I've watched a video or virtual seminar I've done before and just wanted me to explain what I do and how these things all come together to help us to become really effective physician scientists in the realm of cutaneous lymphoma. So this is a very unusual disease that really requires both immunology as well as genetics to really understand how the disease develops, how the disease changes over time, and why you respond to certain therapies. And really, I think most of the people that your doctors, as well as people associated with the foundation, really have to marry these both two different disciplines. How are the T cells or B cells behaving normally? How do they change during lymphoma? And what are the molecular aberrations that occur in patients? So just wanted to reiterate that when I was a resident, as a medical student at Yale, there were a number of real world leaders at the time who were our professors and chair people, and really I just became fascinated by this disease because of so many aspects. But one of the things that really struck me was how there's a unique burden for a lot of patients. It's very difficult to experience this kind of disease when you see it on your skin. I know it takes a mental toll on people, as well as it can really be difficult to deal with, especially because it can be very lonely because of how uncommon the disease is. And then I really thought there was an unmet need for where we can really make a huge difference in how patients are treated. And I really thought that everything was pointing on the way up, and that if we focus all of our energies in the right direction, swim in the right direction, we can really make a huge impact. And I think that it's really a pleasure for me to tell you that some of our work, but really the field, has moved ahead, leaps and bounds from when we first started. And I think we'll be able to continue to deliver better and better care for the patients as time goes on. So what I want to start with is I made this slide about 10 years ago, and this is specifically for Sézary syndrome, but it's just an example. And what I'll say is that a lot of the tests that we do for patients actually have been around for 20, 30 years. And so one way you can look at it is that, oh, it's so great, like we have these tried and true tests. But the other way to look at it is that we haven't really progressed enough to be able to use new technologies to improve the way that we treat patients. And so one of the things I started to do when I first became a professor is I really wanted to say is we know patients are different from one another. How do we take advantage of this information to better understand and treat people? And so our future goals are really to take this same schematic, take a patient, do standard of care molecular test, and instead of using treatments based on physician preference, that we can really determine what's going to work the best for our patients. And so this is going to require continued innovation, both in the laboratory as well as in the clinic. But I want to try to give you a flavor that everything is pointing in the direction that we're going to be able to do this actually very soon. And we need to continue to work together across patients, physicians, and scientists, as well as government and the foundation to move this forward. So these are certain questions and challenges that there are about of working in cutaneous lymphoma. So all of my patients ask these very questions. I'm sure many of you have also thought of this. Why do I have this? What should I expect will happen to me? And what do I need to do? And to be honest, it's almost, as I said before, it can be incredibly lonely in some ways to be a patient. That's why the foundation is so incredible in its ability to bring people together and provide the information you need. And as a physician, it's actually also very important because there are some diseases that are so prevalent, such as breast cancer, where you have tens of thousands of patients that you can rely on to make informed decisions. But this disease has obviously many fewer patients. And so it really doesn't have the same kind of data that is required to make these decisions in the traditional way. So we have to think of new and innovative ways to make better decisions for patients, despite the fact that it's uncommon. And I think to do this, we really have to take advantage of the technology that's available to us today. So this is really the take-home for me and hopefully for you, is that what we're trying to do as a field and the foundation, as well as the physicians and the scientists, are to say, if you're diagnosed with cutaneous lymphoma, whether it be T-cell or B-cell lymphoma, I picture here in this schematic that you're in gray. That's the textbook definition. But we all know, you can look around the room or talk to the patients who've been here before, is that not every patient is the same. You have different stages, you have different symptoms, you have different challenges. And so our goal has been to really think about how do we identify what makes each patient different. It's sort of like the colors that you see here, is that we're trying to see what makes each patient different. And this is a really critical information because we can't tell you what to expect for you personally or try to find the best therapy for you unless we know what makes you different. And we're continuing to work as a field to sort of build this out. But I will say, just as a tremendous amount of pride, in the last 5 to 10 years, there's been really tremendous progress in being able to identify what makes patients different. Now, the second step of this, which is even more important actually now, is if we understand how patients are different, how do we take advantage of that to improve clinical care? And the idea here is that we really have to think about, if we identify that you're blue or yellow, how do we give you the therapies that are best for blue or yellow people? And this part is the part that I think that's going to be the future of this field in the next 5 to 10 years. And really takes a village, patients, patient advocates, physicians, as well as scientists, as well as the foundation and government bodies to help support this kind of work. So I just wanted to say is that one of the ways that we've overcome this is that we've tried to say that every physician has a number of patients. I come from Chicago, where we think we see the highest number of patients in our city, state, and potentially in our Midwest area. But even that is not enough to make enough of an impact. What it really requires are like national and multinational collaborations to be able to learn from as many patients as possible, so that we can say this is not what we think, but this is what we know. And so one of the things that's really critical, and one of the things that Susan and the foundation does really well, is to connect us together as the medical professionals and the scientific professionals. Because the first step of any project that we do in our lab or other labs is to really build a syndicate of institutions that will work together so that we can make the most important and broadly applicable findings for patients. And then we utilize this information. This is just us as an example, but many other institutions do the same thing. We're trying to do is take all this information that we can learn from our patients and then try to use this to be able to make conclusions that could apply to everybody. And then really it's important to have this kind of research syndicate to be able to make findings that are applicable to people from the East Coast, West Coast, Midwest, and the South. And then this is kind of both intentionally busy, but also try to make it very, hopefully, simple schematics. And the thing that you might hear a lot now about is how computers have gotten so advanced. You have machine learning and all these different things. And so we have taken equivalents of hundreds of terabytes of things that we learn from patients. We put them through these different computer algorithms and these basic machine learning principles. And what we try to do is we try to find the story behind each person and what makes them different. And this is a real challenge. It really requires a number of very intelligent young people who are the graduate students, the medical students, the fellows, and the junior faculty that help us to analyze this data. So I just want to reinforce a couple of really important schematics to think about your disease. The way I think about it is it's so complicated for two different reasons. Number one is you might say, okay, we all have cutaneous T cell or B cell lymphoma, but T cells and B cells are incredibly complicated. You need your immune system to fight off viruses, fungi, any kind of infection that you could possibly have. And your immune system has evolved to be able to be incredibly nimble as well as specialized. So you have antifungal T cells and B cells, and these are all different from one another. And these different cell types actually all can become possibly cancerous. And so for us to really understand, the different lymphomas, we have to understand the complexity of the cells that they come from, the T cells and the B cells that they come from. And then superimposed on this is just the fact that all of us have half of our DNA, our genetic material from mom, and half of us from dad. That's how, that's the genome that you're born with. But it turns out that the cancer cells have two genomes. They have the one you're born with, but they also develop new aberrations that you were not born with. And these are really the first events that actually support the first steps for you getting the disease. And so you have to understand both of these concepts that are actually being incorporated into clinical care to be able to understand what's going to happen to you as we move forward. So this is our model, that you have different types of cells. This case is a T cell for patients with mycosis fungoides. And there's many different mutations. And these cause the elaboration of molecules that give you the rash, the itch, everything, et cetera. And this is why this is such an unusual disease and requires a very specialized research program to go forward. So I'm not going to go into this in really high detail because it's very complicated. But I just want to say is that repeatedly we've found that the different cell types make a big difference. These are examples of gamma delta T cell lymphomas, again, another type of cutaneous T cell lymphoma. And it actually turns out that there are many different types of cutaneous gamma delta cells. They could be blue cells or red cells. They're called delta 1 and delta 2. And it turns out that we've learned from research through multiple patients in multiple institutions that actually the cell type you actually have that is the centerpiece of your lymphoma is a major driver for what to expect for you. Some patients will have, a complication or symptom A, and that'll be because you have a blue or delta 1 lymphoma. And then some people will have complication B, and that'll be because you have a delta 2 lymphoma. So these are the kind of concepts that we're going to continue to refine as we move forward. I'll just skip through this. So I think as a field, we've really made a lot of progress on the genomics. We've been able to find the molecular defects in each of your cancer cells and put this together. This has really started from about a couple dozen patients. Now we've had hundreds, and we're getting close to 1,000. And this kind of research has enabled us to really get a fine GPS-like map as to what makes each patient different. It's now going to take us time to figure out, now that we know the differences, what does it mean for the patients and how can we take advantage of it? And I just wanted to show you some stories that are inspirational for me, and I hope that will be somewhat inspiring for you to think about how this has really been able to not only affect how we study the disease, but also how we can potentially treat the disease. Okay. So I'm going to first talk about here is, before I get into the patient stories, is that we really have been thinking about how to even move this further. So a lot of the technology that's been done, and we've made incredible progress, is doing what we call bulk RNA-seq. And you don't have to know the term, it's very technical, but I'll try to explain it to you. What it does is you take your patient samples and you get molecular tests. This can now be done in the clinic. And so there's many different labs that can help you to do this as we go forward. But what I tell people, and especially my patients, is this is kind of like having a smoothie. And it's like, you can say, oh, I taste a little blueberry and I taste a little mango and this is what I have. But it's been really able to help us because we do get a flavor of what each person's disease is. What I wanted to tell you is that the way that the disease is going to move forward with research, I think both from, with all the help of all the funding bodies, is we've now gotten this ability to actually get single cell resolution of both the tumor as well as your immune system responding to the tumor. And so if the bulk RNA-seq, which you can do now with your patient samples, is kind of like a smoothie, I think of single cell kind of being like a charcuterie board. You can imagine you know exactly, I know that I have like manchego cheese and, you know, like salami from, or a sausage from Poland, and you can utilize this information to even get even further. So before you had this, you know, people who had different diseases, but you really couldn't understand why they're different. We now have the smoothies where we can sort of get a good idea of the flavor, and we can really think about the precision it takes to do this. And this is the kind of thing that's going to be the future in the way that we understand and treat your diseases. Okay, that's what I was trying to say here. So I think the main thing that I always think about is all of our research, as for myself and for the community, is really patient-focused. And so I think the two areas that are going to be the biggest impact are, one, is how can we tell patients what to expect? You know, how do we tell patients you're doing well or not doing well? This is part of the, you know, the angst of being a patient with this disease. You always look at your skin and you're always worried that this is going better or worse than you think. And I want to be able to provide additional information to say, I think you're doing amazing and we're continuing to do great. This therapy is perfect for you. And so we have to do this is to utilize that kind of information we had before, the smoothies and the charcuterie boards, and try to link it to the different therapies that you could possibly receive. And that's going to be the critical, I think, next step to how we move forward. But the other part, which all of you I'm sure are interested in, is how do we develop new therapies? This is an uncommon cancer. It's very difficult to get the patient cohorts to enroll for all these different things. How do we learn as much as we can so that we can give new therapies for patients when they need it? Sorry. And I will say is that in a lot of ways, the future is now. So I just wanted to show the schematic, and I'll give you an example, is that we now have these ability of using these multinational, multi-institutional syndicates, where a colleague of mine from Boston, from Harvard Medical School, will be able to analyze a patient sample, and we go through this molecular analysis or DNA RNA-seq, and they'll call me and say, hey, we found this mutation, and we don't know what it does or what to do with it. And actually, now we can actually model this, put this into a dish, and what we're trying to do is say, hey, a clinical trial may take 5 to 10 years to develop from inception to completion. But can we take advantage of drugs that are already available in the clinic, maybe for other diseases, and then use them for this kind of approach? And so we are actually trying to do this in real time, so that if we have patients who really need it, that we can sort of find the molecular defects and say, hey, can this predict that you'll respond to something that actually is available in the clinic? And so this is just one example where my very talented graduate student, Julia, was very interested in this case where we got a call from a hospital. I won't say where it is just to keep patient anonymity. I'll just say it was a pediatric patient. It was a young girl who developed this molecular defect. It was never seen before in this disease. And so we said, hey, let's try to see if we can identify, you know, what it does and if we can possibly find a therapy. And she's been working through this actually with these very interesting cellular assays and been able to try to find drugs that actually work for this patient. And it turns out that this patient received this drug in part because we were able to do these studies, and she's now doing actually very, very well. And so we have another patient we were called about, is a 90-year-old woman. Again, I just will try to not talk too much about the personal details, but also the clinician had a confusion about what to do because we don't have therapies for all the different types of CTCLs. So we did molecular analysis almost at the point of care, and we were able to generate a test that pathologists can do that can help us to identify what to do next. And we have been able to utilize medications that were developed for other diseases. And this patient who was 90-year-old and really in need of something that would work well for her, we were able to give her a medicine that's actually really well tolerated, but developed for another disease type. And she had about 90% regression of her lesions as well. So I just want to say is that I really am encouraged and inspired to move forward. You know, I think that we have a number of the brightest minds in medicine and science thinking about these questions. And we're really trying to put together the charcuterie board for each person so we understand who's blue and who's yellow. And we're using as much innovation and scrappiness as possible to try to find these therapies. You can imagine that we're really here generating N of 1 trials, like one patient, one trial. Let's do the best we can. And if things work, we can sort of think about how to apply this more broadly. And I just think for me, it's just an inspirational time because, like I said, for 10 years ago, I made this slide that we sort of treat patients the same way that maybe it's been treated for a while. But now we'll be able to really in real time, predict new therapies for patients as they go forward. So what I want to say is that in the landscape of clinical trials, which my colleague will also go into, is just that everything that we're doing is really targeting either the T cell or the B cell that is causing the disease, the T cell or B cell in the cutaneous T cell or B cell lymphoma, or the mutations, which are these molecular defects that we describe. And just like I said, that combination is what gives you the disease, and those are the things that we're targeting with different clinical trials. And so you can imagine that there's therapies you can develop to really target different types of T or B cells. And actually, these are being tested right now through many different mechanisms to be able to kill the cell that actually causes the lymphoma. And then the way that your mutations or molecular defects work is that your DNA encodes RNA, which encodes proteins. And this kind of process actually is mutated and taken advantage of by cancer. And what we can do here is also we can have multiple different types of clinical trials that can target each and aspects of these. And again, the future that I envision for all of us is that it's not going to be a clinical trial where we test 10 people who are gray. We're going to test people who we know are blue, yellow, or red. And then at the end of it, we not only know who, does it work, but also who should receive it, and who's the patient who really needs to receive it, okay? So as we talked about this before, I think these are the challenges of dealing with an unusual disease that has unusual symptoms and that makes patients' lives, incredibly difficult. We have come up as a field with different strategies to work together to be able to try to answer these questions. And my hope is that if we do this 5 to 10 years ago, we will no longer have these questions. We'll have new questions, which is like, how do we move on from here? What do we do next? And these are the things that we're hoping to answer in the next few years. So how do we get there? I just really want to say is to emphasize is research takes a village. For me, I have a lab of about 10 people who are graduate students, medical students, MD, PhD students, and postdoctoral fellows. They have to all do each of this work individually by hand. And, but they are critical for the breakthroughs that we're seeing in the clinic that we're trying to implement. But also they're critical because you want the next generation of the physicians you see to be inspired by this disease and think about what to do forward. And really what my experience has been as a teacher is that a lot of people get really inspired when they can go really deep into something and learn about it and see how they can make an impact. And so research is really a critical step, not only making patients' lives better, but you want to have future leaders who are engaged, intelligent, recruited, because they are very invested in helping patients. Okay. So, sorry, this is a slide I took from before. Our goal is really to cure patients, and I think everything I told you is really in line with our mission. This is a picture I took with one of my patients who was convalescing a few years ago. It was taken by my daughter on a Saturday afternoon. And I just want to say is that everything we do is really designed to improve therapies so that the next patient that comes in will be able to have a much easier time and a much better outcome than the ones we treated in the past. So happy to take any questions.
Thank you, Dr. Choi. I hope everybody is like as excited and inspired listening to this presentation as to where we are, how far we've come, and what the future looks like. And to your point, it takes a village, right? It's all of us rowing in the same direction to really make a difference. But know that, and I always have to share this John Zick quote, Dr. John Zick from Vanderbilt, because at one of our programs many years ago, he said, know that there is someone in a lab somewhere in the world with the light on working on your behalf. And I really do believe that's true. And it is an international community that is working together to try to bring this to reality. And you just had a little glimpse today of some of what's going on behind the scenes. So thank you so
Thanks
much.
so much.
And I know Dr. Choi has to leave us here. He's just in and out. So if you do have any questions, we'll take them now before we go to our next presentation. Yeah.
Dr. Choi, how do you go about getting the molecular test done?
Oh, that's a very, so I'll say is there's several aspects. So number one is obviously, if you get the molecular test done at your physician's office, there's an interaction with the insurance company. So that is, well, our challenge in the future is that what we want to do is justify to the insurance company that we can get the molecular test, because it actually will change patient care. But right now, these molecular tests are sometimes paid for insurance, sometimes by not. So I would say first route is if you can get this done by your physician. There are test labs that can do it, either in the hospital where they practice or national labs. But I will say the challenge right now is the insurance. So this is part of what we need to do, is to take things that look like research and actually implement them into patient care, is that we need to be able to offer the molecular test and have the insurance companies cover it, because we show that it's useful. And this part is not completely done yet. Yeah.
Where would you find the people in the hospital who are doing the molecular? How do you know where to go?
So I think that for the large part, your physicians will probably have some idea if you ask them, but also we can, if they reach out to me, we can help to sort that out. Yeah.
We have a question from the online audience, if we can take one from them first. Are these techniques appropriate for someone with early stage, low-level disease to try and get ahead of the disease instead of waiting for progression?
We don't know the answer to that yet, to be honest. And I think that what I'll say is, if everyone gets enthusiastic about having molecular tests on the disease, I totally understand that. But we're not ready yet to be able to implement for every patient. We've been learning a lot, especially from patients who have limited options with standard of care. So I would say is that I don't think we can do that now, but I think there's a time in the future where you could do a test and be able to predict who needs to have more intensive care despite looking like everything is going well. So that's a very important question. We're not able to answer it now, but it's one of our goals. So yeah, there's a question there.
This is how I get my exercise for the day.
Thank you for that great presentation. So obviously at the end, the last slide, which is most important probably to most, is a cure. So, and I appreciate all the specifics about how you're going to get there. How far are you, and what type of timeline, if there is one? I mean, I know I'm asking probably unanswerable questions, but be interesting to know what your expert opinion would be on where you think you're at, or where you think the group is.
I think that we're well along our way. If you ask me, without any changes in technology of the field, it'll still take a couple decades, I think. But what I think you have to think about is you can't stop science, technology, and biology, I actually think. So I think there's going to become a lot of new tools where this could possibly happen in 5 to 10 years. But I think based on the current progress, it'll take longer. But you have to build in that there are scientific innovations that totally change the game. Just like the smoothie methods, that didn't exist 20 years ago. But now we have this incredible insights, and that really pushed the field forward. So I think those are the key. But I will emphasize that we have to inspire the next generation of leaders now who will be continuing to do this fight in the future. And I think that is sort of related to your topic, but not exactly. We're going to try to cure the CTCLs or the CBCLs, but we also need the people to fight it in the future. And that starts now. And that takes 10 to 20 years, which is very important. So.
Hi, Dr. Choi. I got kind of two-part question. First is, I feel like what you talked about with precision medicine and individualized treatments and care is so critical with this disease because we all have a totally different story. And so I feel like, and then one of the challenges with this being a rare disease and all the FDA approval process of getting anything done, it is very difficult to work through clinical trials and those type of things. So my first part of my question is, as you move into this individualized, personalized medicine world, is the FDA moving that way and allowing you to do clinical trials with small you talked about the one person thing, it almost has to be very small groups in our particular disease. I don't know how you could do it with big groups. And yet the FDA and all the approval processes for that, is very difficult. So that's part one of my question. Part two is stem cell. There's a lot of stuff going around the world with really innovative stuff in the stem cell world to cure a lot of diseases and fight a lot of diseases. And obviously we've had the total, the cure on the extreme side with stem cell, with a bone, whole transplant. But is there any work, I didn't notice stem cell at all on your slides. Is there any work being done in that area in particular that moves the bar on even able to get stem cell injections that maintains, as a treatment method for just maintaining the disease versus the whole transplant only when you're in an advanced stage type of thing?
Yeah, so to your first question, I will say is that, as physicians, just seeing things that happen in cancer in general is really critical. So you may have seen the data from a few months ago where they treated people with colorectal cancer in one trial and 10 out of 10 all got cured. And so I don't think the FDA is going to be like, well, 10 out of 10 got cured. Who knows if that's, that's a signal. And I think one of the biggest challenges here is if you ever read the literature, they enroll all patients with lymphoma and they almost use no information about what makes people different. So I think the way to overcome the challenge of people is not to do something we can't do, which is get more patients. We just have to know which patients we're enrolling. So even if we see a signal in a subset of patients, we know that for them it works better. And so I think that this kind of characterization is going to be a critical should be a critical complement to all future clinical trials. So we can interpret, who does this work for? Where does this work for? So if we can do a very targeted clinical trial, and instead of getting like hundreds or thousands of patients, we can get several dozen that are the same color, and then that's where it works. That is much more feasible, in my opinion, than getting like hundreds or thousands in general, I think. So I think that's number one. And number two is that I didn't mention the stem cell transplant, but I think this is part of the complexity of why we need more research. Each of these will require a young person to think about this idea and study this particular therapy. How do we make it better for which different patients? How do we make this therapy better for which different patients? I just mentioned 3 or 4 different therapies. If we had stem cell transplant, and we're going to talk about other immunotherapies, each of these will require a village in each of the therapies. So I think your question is very important and something we have to deal with as a field, but it also reflects how much need there is actually for more and more research, because it's going to need more people to study all the different things that we want to try, I think. So.
Choi, I have one from online. If you could just repeat it so that you hear it.
Okay.
Can you tell us about any clinical trials you are currently in right now?
That I am currently doing right now?
Yes.
We talked about these patients with these CD8 T-cell lymphomas that have these JAK mutations. We have a clinical trial at Northwestern where we're trying to test the concept that even with 5 to 10 patients, we can basically cure this otherwise very difficult to treat disease. And so that is an example of one we have at Northwestern right now. We're using these molecular data to guide us.
Thank you for being here, Dr. Choi. My question is, do you see in the future, however long that might be, that you'll come in, you'll be diagnosed, and the doctor will say, well, you're blue, you're green, you're whatever, and that will drive the care that they provide?
Yes, so I think you'll learn that there's 2 types of researchers. Many patients haven't seen researchers, but there's 2 different types. So one type will explain how the disease is so complicated that you have to be a super, super expert to be able to understand that. But for me, that's not the goal. The goal is to make it simple. So the goal is that, yeah. Anyway, my vision would be is that right now you need extreme.
You can talk into the mic.
Right now you need extreme experts with incredible expertise studying the disease for many years to understand all the nuances that are appropriate for you or your other patients. I guess the vision that I have, at least, is that we can have a computer printout that will tell like 99% of the information, that can really uniform and make the prognoses, the diagnosis, the staging, and the treatment decisions more uniform and data-based. So it could be exactly like you said. There could be a test that can tell us you're blue and you're green, and this is more likely what you should do. I think that's the kind of thing that I would hope for in the future. So.
I think we'll do one more here, Kelly.
I'm honored to be able to ask a question. Thank you so much for that really inspiring presentation, Dr. Choi. My question is around how do patients find that pathway into getting involved in research? Because I don't have lymphoma, but I represent a community where we've been working on something for 10 years, and we're finally in dialogue with the NIH to run a pilot study on this. I think there would be so much interest in the patient community to get involved in these research projects because we all want to know. We all want answers. But how, like, what is that pathway to find a research center, a study, somebody at NIH without having to go through 5, 10, 15 years of making that connection? So a lot of times we don't have the time in the clinic to talk to our doctors about this. We're just getting treatment. So what is your recommendation to the patient community to try to kind of bridge that gap?
So I'll say is that I think most of the physicians who treat this disease are very interested in research because they want to improve the way that their patients will be treated. So I think if you go to an academic center, there's a lot of opportunities for you to do this locally. But I actually think that one of the key aspects here is that a lot of this actually takes resources and funding. So one of the things I would encourage people to do is if they really want to make an impact, it may sound very mosaic and very mundane, but actually, being able to think about how to participate and raise money for to pay for the people who are doing this kind of research is, I think, one of the biggest impacts you can make. So I think as a patient, you can participate in your local studies. I will say that we are trying to create a general way that patients can just send us their samples, but this is actually really complicated right now. But this is maybe something in the future. But I think that one way you can get involved, besides having your own disease be studied, is actually trying to help support this village that is really required to push this forward. Because I want to have enough, smart young people answering all the individual questions here, but each of them will probably require 1 to 2 or 3 different people. And then we need resources to help pay for their salaries as well as their very expensive studies. So I will say in a mundane way, if we raise funds at every level, foundation, you can also talk to NIH or the government about raising more funds for cancer research as well as for this particular field. All these different things make a huge impact for us who are trying to do it. So.
Great. I think that's great. And Kelly and I, we can chat a little bit about that on our session. And I think also, thank you, Dr. Choi, for everything. And, let's bring in the youngsters so they can carry this forward, right? That's what we're all going to try to do, is really encourage the next generation and to be trained by these brilliant folks to keep the momentum going. So thank you. And
thank you very much for coming.
And now we're going to turn it over to Dr. Stefan Schieke, who is one of our local cutaneous lymphoma experts here in Washington, D.C., also a researcher and innovator in cutaneous lymphomas, working collaboratively with the rest of the researchers around the world. And we're going to talk a little bit about, he's going to talk a little bit about clinical trials and precision medicine, kind of add on to what Dr. Choi kind of set up. and give you a little more insights into some other things that might be happening. So I'm going to turn it over to you, Dr. Schieke.
Perfect.
Thank you for joining us.
Susan. Thank you very much. Good morning to everybody. Thank you for being here. Yeah, it's a great opportunity actually to tell you a little bit about the things that are really upcoming and new. You've heard the presentation by Dr. Choi, which was a great start, and I'm actually going to take it a little bit back and want to think with you together about what does that all mean for clinical trials? Are the clinical trials we are doing right now actually set up in a way that we can answer those questions, that we can find these precision treatments? And at the very end, I want to go into details and explain a little bit about what are immune checkpoint inhibitors, what is that immunotherapy, and the other thing which you might have heard before are these so-called CAR T cells, relatively complicated biological backgrounds, but I want you to be able to understand it because I think that's where actually a lot of the future lies. So before I do that, I have to figure out how to advance the slides. I want to briefly introduce our team of cutaneous lymphoma clinicians at the MedStar Georgetown Cancer Institute. We are currently housed at the Washington Hospital Center. And they are great colleagues. Arthi Shenoy, you can meet her tomorrow. She's part of the Q&A session in the morning, radiation oncology, but really very, very important colleagues in dermatopathology and pathology. And all of us together are trying to really, not only diagnose the disease, but figure out treatments. And Dr. Choi is right, the easiest access to clinical trials is actually through your local lymphoma docs. Not always do we have all the clinical trials open at our site. That's virtually impossible, but usually we know something about this or that and then can determine together when would be the right time to go there and contact those people. All right, so what I'm presenting you, and also similar to Dr. Choi, why do we talk so much about precision medicine? Well, because, just a few weeks ago, we had the United States Cutaneous Lymphoma Consortium, USCLC annual workshop, and it was one of the times, a few years back, we had that too, where we focused on precision medicine, because this is really the future. This is medicine 2.0, or you might want to even call it 3.0, but this is really where the future lies. But as always, when you think about the future and want to move things forward, there is a challenge, and the challenge is actually quite significant. This is a little bit overlapping what you have heard before, but since the topic is so critical and central, I think having the same information from a second person here, I think is actually helpful. So why is it challenging, precision medicine, and what actually is the problem? The problem is that cutaneous T-cell lymphoma, and you can also say cutaneous B-cell lymphoma, is a very heterogeneous disease. And heterogeneity in this case really means that it can be very different between patients. We call that the inter-individual heterogeneity among patients. We have patients with just patch stage disease, plaque stage disease, something we call erythroderma, for example, Sézary disease, where the disease is affecting the entire skin. But there's also tumor stage disease, and all these are different stages, different almost variants of the disease. But to make it even more complicated, you can find these different types of lesions, or almost these different variants of the disease, not between patients, but also in the very same patient. And that is the so-called intra-individual heterogeneity. And that is very important to keep in mind, that the disease looks and behaves different, not only among patients, but also in the very same patient. And we often see that a lot, that we have very mixed responses to treatments in the very same patients. It could be that some spots respond and others just don't respond. And there you go. Then we have to figure out, why do these single spots not respond? What is going on there? And to make it even more complicated in terms of the heterogeneity, The disease, as you know, does not only affect the skin, but it oftentimes involves the blood. Of course, Sézary syndrome is defined like this through this enormous leukemic burden, but even in patients with sometimes early stage mycosis fungoides, we can find cancer cells in the blood. It can spill over into the lymph nodes. It can also start to involve visceral organs. So, and all these compartments don't have disease that behaves exactly the same, but all the treatments basically have to cover these different compartments. So in here, I'm showing you the so-called, this is basically what we get back from clinical trials nowadays. And everything Dr. Choi said and everything I'm going to show you, even though it makes a lot of sense, but this is not how we are doing it right now. What we are basically presenting is where the field will move to. How are we going to do this? And the exact timeline is hard to tell, but I would think some of these clinical trial modifications will probably happen within the next few years already. But this is what we are getting back today. We get these progression-free survival curves, and we start with 100%, and every time the curve drops means that one patient or more than one really have a progression or they unfortunately passed away from the disease. If you're under the curve, well, that's great. That means you're responding, because at the very end here with this one treatment, which is actually brentuximab, there are still patients alive. If you're above the curve, that means your disease is progressing. And that's how we arrive at these numbers, that bexarotene has a response rate of roughly 30%. Well, that's not great, right? If you look at every single patient, it's really binary. Either you're going to respond or you're not going to respond. The 30% is in a way meaningless for you as an individual and also for us as providers, because I want to know, are you going to respond or are you not going to respond? Are we wasting time and money and energy and mental energy too? So what we have to figure out is what really defines the responders and makes them distinct from the non-responders. And if we can identify all the responders, well, then the response rate would be 100%. And this is unheard of. And this is the idea of precision medicine. We want predictors that allow us to figure out, well, you should get this treatment or that treatment, and your response is 100%. You are going to respond. It's not a 30% chance. It's not 0. It'll be 1. That's the idea. So, and at this point, we are not there yet, but we are getting there. And one of the reasons we are not there yet, but I wouldn't say, or I wouldn't think this is necessarily negative or pessimistic that we are not there yet. The good thing is we are currently learning and understanding that our current approach and our current clinical trial design is inadequate. It has been okay for several years, but we can do better. And that is because, the basic biology tools we have in the lab provide such a deep insight into the disease that at this point we have, as clinicians and clinical trial and study investigators, have to do a better job to keep up with the basic biology. We have to take a step back and think about how to design the clinical trials that we actually are able to use the information that we get from the lab. So, and here, and Dr. Choi already mentioned that, you have patients with the same disease. Let's say this is cutaneous lymphoma here, but the different colors, he called those the different smoothies, right? But very, very different, distinct, maybe molecular patterns that make these patients different. But in the clinical trial and in the response curve I showed you, they all get the same treatment. And of course, some will show an effect, the treatment is going to be great. Others, there's no effect, we call that stable disease. And others might have a lot of adverse effects, side effects, or the treatment might simply not work. So what we really need in order to figure out in which group every individual is going to be effect, no effect, adverse effect, we really need individual diagnostic, prognostic, and also therapeutic response parameters for every single patient, because we know there is just no one-size-fits-all treatment for cutaneous lymphoma, for not only cutaneous lymphoma, any disease, any type of cancer. There is just no one-size-fits-all treatment. Otherwise, there wouldn't be hundreds of drugs to treat hypertension, for example, high blood pressure. You know, this is actually very, very tricky to treat high blood pressure effectively. It is not less tricky than treating many types of cancer, including cutaneous lymphoma. So we need to be better to figure out what makes you special and which special treatment do we need for you. And I would definitely say at this point we have all the tools. We just have to put it all together, and we are doing it in small steps. So we are absolutely on the way to solving this challenge here and dealing with it. This busy slide here is introducing what you all know, but specifically in the field of medicine, big data. Whether somebody is tracking how you use Google, which is big data, or what you buy on Amazon, or you throw everything together, what we put in health records, what the pathologist figures out in terms of what type of cutaneous lymphoma a patient has. As Dr. Choi mentioned, the sequencing data, the genomic data, or the response to drugs, we can put that all together because we have the data. It's already there. But then the challenge is to retrieve actionable information out of this data. And that's where a lot of the research is going on right now, and this is not done by biochemists or biologists, people like me and Dr. Choi. This is really done by mathematicians, informaticists, people who can design algorithms that allows us to really distill actionable, relevant information out of this ocean of data. That's the future. You know, a big part of that is also how we, you know, the charting we do. You go to your physician and you might see them typing in the computer, or maybe the resident or the fellow is typing into the computer while the attending is talking to you. But sometimes, you know, even extracting the data out of the computer and synthesizing it all, that's also one of the challenges we are dealing with nowadays. But much easier than accessing and synthesizing the clinical data, we always have basic science, we always have the basic biology. It is actually relatively simple to get a skin tissue, get cells out of it, extract DNA, extract RNA out of it, all in the lab on a research basis, and then we have all these omics, right? You've heard about genomics for sure. There are metabolomics. That's what we are doing, where we focus on metabolism. Which nutrients do these cancer cells actually need? Which nutrients do they need in order to resist treatment or to travel around the organism? How can we attack the metabolism? There's immunomics, there's epigenomics, proteomics. Which proteins can you find in these cancer cells? So this is all the stuff we are doing today for many types of diseases, for virtually every single type of cancer, but also for cutaneous lymphoma. This is happening. This is absolutely there. And using that information and distilling actionable information out of that is very doable. This is part of that, you know, gave rise to the clinical trial, which Dr. Choi introduced, but other druggable targets will be defined through exactly this approach in the coming years. And there we are not talking about a 10 to 20 year timeline. I think here this, you know, the next discovery might just be around the corner. You just don't know. And if we have that, you know, then we can actually start thinking about precision clinical trials. clinical trials that match our biology information, our biological insight into the disease. And I just termed that clinical trials 2.0. I haven't read it somewhere else, so I don't think it's somehow protected. I'm using it here, but I'm not claiming that I invented it. So clinical trials 2.0, this is exactly what we have to do. We have the patients with the very same disease, cutaneous T-cell lymphoma, let's say maybe Sézary, maybe mycosis fungoides, but what we want to figure out, what makes them unique on a molecular level. We can do that with genomics, we can do that with metabolomics, we can do that with immunomics. We can look at basically every single biological feature of the disease and try to categorize it, try to figure out where is a unique defect that actually causes maybe the disease. And that's really what we try to figure out, right? In biology of the disease, cancer biology, we are trying to figure out what really causes the disease. If we know that, then we have a really great treatment target. So, and then we basically group the patients. We say, okay, there's a mutation or there is some dysfunction in pathway 1. They get drug 1 or 2 or 3. And then we are at the point where we actually all would love to be right now, that actually everybody has a positive effect of the treatment. And here's a little bit more theory, because this shows you that there is actually active research and active thinking going on how we restructure our clinical trials at this point. And there are a few different ways, but I think these are actually very instructive and interesting to look into. One precision clinical trial approach is called the basket trial, and the other one is the umbrella trial. But what you can do is you can have multiple different cancer types. It could be breast cancer, it could be pancreatic cancer, it could be melanoma, and cutaneous T-cell lymphoma. And maybe all of them share this one mutation or this one thing that makes that is different, but different in all of them. And maybe we have a drug that can target this critical issue. And that's how we basically can lump different cancer types with the same molecular defect and then treat with the very same drug. And the advantage here is that, well, we can focus maybe on a molecular defect which we can target, because we might be able to find a molecular defect, but it is not guaranteed that we actually have a drug that can target it. So that's one approach. The other one is the so-called umbrella type. You have patients with all cutaneous T-cell lymphoma, and then we figure out, as Dr. Choi described, I described it before, as well, that different gene mutations, for example, or different metabolomic features. Maybe this cancer cell needs glucose to grow, another cancer cell needs another metabolite, certain amino acids to grow. We focus on this and give all these patients different drugs. But everything, again, is really, and the key here is biomarker driven. And I can guarantee you this is happening. There are, not yet for cutaneous lymphoma, but there are for systemic lymphomas in the hematological cancer community. There are clinical trials that follow exactly this design that are going on right now. But of course, it's always good to be a little bit cautious and figure out what problems are, because we do need to identify biomarkers that are druggable. It doesn't make sense to have a biomarker and we don't have a drug. It's still a good finding because we need to then find the drug, but in order to turn this into a clinical trial, we need druggable biomarkers. The other thing is that we can sequence all day long, but we might not find actionable targets. When I was in medical school, and that was really the end of the 1990s, early 2000s, people were so excited about the genome sequencing and said, we have everything solved now. Combine that with the stem cell biology, everybody was super excited and said, we're going to solve cancer. We just can solve myocardial infarctions, heart attacks, because the dead heart tissue will replace it with the stem cells. Many years later now, more than 20 years later, I think that part of stem cell biology in medicine has disappointed. It is not that easy. It is not going to work this way. The sequencing, I think, as you have seen before, is actually a little bit more promising, but it's not the only answer. The other thing is, and given that we have all these other omics, I think this is not a reason to despair. It is just, we all have to keep an open mind. The answer is not always in the DNA. The other thing is that we have limited pharmacological options or tools, medications, but with ever-expanding list of targets, we will also have an ever-expanding list of chemical agents that we can use as drugs. This is all very expensive, and I can tell you right now, a lot of the reason why this is an N of 1 with one patient right now is also usually a cost issue. Because on a research basis, if we get a regular research grant for just over $1 million for 4 years, we can easily treat a few patients. We can do all these omics for a few patients, but we can't offer that yet on a standard basis. But here I would be very optimistic because all these metabolomics, genomics, the sequencing used to take extremely long, and it used to be very pricey. The price has dropped, and the time it takes to run these assays also has dropped dramatically. And you know, that basically brings the other thing up. We need to now start working on bringing these research tools into routine clinical practice. And that does involve the FDA. That does involve not just 1 or 2 labs figuring things out. This does involve the community and also a lot of regulatory points. And lastly, we need more refined big data analysis tools because, like I said, the data is there. But what people are struggling right now is to figure out the exact perfect algorithms that actually give us something we can work with out of these data. So I put this here. These problems are all solvable. And that is important to keep in mind. And we are actually working on this at this point. So after this overview of how we will structure clinical trials in the future, which I think is actually a very optimistic perspective, I want to tell you a little bit more about 2 types of immunotherapy which will actually probably become very, very important going forward. One are the immune checkpoint inhibitors, or simply immune checkpoint inhibitors. You might have heard about those. Those were really big in the news several years ago. But it started all with not lymphoma, but another type of skin cancer, melanoma. And here, this is one of a nice picture, actually, a biopsy of a patient with melanoma. The melanoma cells are these cells here with a dark nuclei, look very angry. This is the cancer. But then you have these little guys around it, the small round nuclei, and those are the lymphocytes. And what are they doing there? They have realized something is wrong. We don't like this. This cancer is just not looking good to us. We should fight it. But the cancer is smart and has figured out a way to calm these lymphocytes down. They get there, but then they're basically put to sleep. They stay there. And what these immune checkpoint inhibitors do, they take the brake off. They wake up these lymphocytes, and they can get activated and destroy the cancer cell. And I want to get even into a little bit more detail because there is one drug that's called pembrolizumab. And for that, we need to understand how we can activate a T cell and how we can actually put it to sleep, suppress it. So for activation, what usually happens is that a T cell has surface receptors, right? Proteins on the surface. They need to bind to other receptors on the cancer cell, and that tells the lymphocyte, the T cell, well, this is a target, I need to start killing you. But so this doesn't happen all the time. Nature has built in with the cancer cell, because cancer was not really how this all started. The idea is a T cell is not supposed to attack your own healthy tissue, right? So a brake has been built in, and that is so-called PD-1, PD-L1 interaction. If these two proteins bind together, the signal to the T cell is, no, 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 don't do anything, I'm a friend, don't kill me. What's happening is that the cancer cells are using that very same mechanism to evade the destruction by the host response, by the lymphocytes. So what can be done here, and unfortunately, yeah, here, you know, there were antibodies. There is a drug that actually binds to these, prevents this suppression signal, and that's called pembrolizumab, and you will hear more about it. That is the immune checkpoint inhibitor. It basically makes sure that the cells that got recruited to the cancer recognize the cancer as something they can fight, the brakes are being taken off, and the lymphocytes are starting to fight. And one of the big, big advantages in immunotherapy, and we have seen that for melanoma, in different types of lymphoma as well, the immunotherapy is the one that actually gives you very long-lasting responses. And that's why people are so hopeful about it. We still have patients who were put on with melanoma on one of these agents, and 10, 15, 20 years later still have a response. Have a response when they were put on the drug, where years before their life expectation was only 6 months. And 20 years later, they are still alive. That is something we have only been seen so far with immunotherapy, and that's where really the big hope lies. And here you can see that is from a publication from Stanford, 2020, about 3 years ago. This is one of these checkpoint inhibitors, pembrolizumab, in mycosis fungoides and Sézary syndrome. Here, 2 patients with a really nice response. You can see it. But here you can see every line is one patient. And you can see the response is pretty mixed. The patients who had a good response responded to the drug, the disease got suppressed, are in blue. And then we have the patients who didn't respond in kind of brown here, and then the gray, stable disease, also no response at all. And this variation of the response shows you again, well, what we need to do now is figure out why did these people respond and why did these people not respond. If we are using our omics and our basic biology tools the right way, we can probably tell you, even before we start the treatment, you are going to respond and you're not going to respond, but we have another treatment for you. And that's the idea. But there's much, much more to come, and I think this is really in cancer medicine, cancer biology overall, over the last 10, 15 years, this is one of the big, big breakthroughs. And the other thing I want to talk about, and I'm keeping the time in mind, which you might not have heard yet, but will much more in the years to come, are so-called CAR T cells. I'm bringing this up here because there are actually clinical trials in cutaneous lymphoma right now. And the other thing is the biology behind those is pretty complicated. I want to take 2 minutes to explain it so that you know in the next 5, 10, 15 years maybe, well, I know what CAR T cells is and what all the hype is. And why this is actually important. So here the idea is, similar to the immune checkpoint inhibitors, the immune cells are actually also smart. They often know that a cancer shouldn't be there, that a cancer is bad, that they have to fight cancer. And then the idea is now, the question is, how can we engineer the immune cells or the T cells to really make them better able to fight cancer in patients. And one way is, well, we take the brakes off. The other idea is, well, let's take those cells into the lab, turn them into super killers, and then give them back to patients. And that is done with what we call the chimeric antigen receptor, or CAR T cell. And there's a great explanatory schematic on the website of the Leukemia Lymphoma Society, which I took because I can't do something like this myself. It looks really nice. But let's walk through this together, even though it looks complicated, but the basic idea is actually not complicated. So what we do in the clinic, and again, this is happening right now. This has been really described several years ago. This is happening with hematologic malignancies at this point. So we do a simple, more or less simple blood draw that allows us to get the T cells. We want the T lymphocytes, which we then take to the lab. And here is the critical part. We change them, we modify them. We put a gene into these cells. In a way, this is mini gene therapy. So we put a gene into these cells, and based on that, because of that, these cells start to express receptors, proteins on the surface of the T cells that can recognize cancer cells. So we basically have kind of mini cancer-finding proteins on these cells, which are not there, which were not there before. Okay, we do that usually in only a small number of T cells, but once they are what we call here transduced, and they express the protein and then the receptor here, we multiply them, make millions of them, then put them back into the patient, and then see what's happening in the body. The cancer cells start binding and recognizing the cancer cell, but because they do this very, very efficiently, they are really called super killers. They are able to destroy the cancer cell, something that the unengineered normal T cell cannot do that efficiently. So that is the whole CAR T cell therapy. And what you need for that is you need something on these cancer cells that can serve as a target, something that is specifically recognized by these super killers, telling them, I want to kill you. And you can use different, I just put them on here, CD4, which is expressed on pretty much all mycosis fungoides cells, CD30, which you might have heard before. It serves also as a target for the drug brentuximab. CCR4, another protein, serves as a target for mogamulizumab. But what we can do now is we take those same targets and make them basically make them recognizable by these super killer T cells. So here are the two clinical trials that are open right now for the CAR T cells. One you can see is actually a combination. There are two structures that are recognized, CD30 and CCR4, and another one is CD4. The biggest challenge at this point, though, and of course it all sounds great, but there's a challenge, but we are working on that. People are working on that. The biggest challenge at this point is ensuring specificity and safety. So specificity means these super killers should not start killing your kidney, your heart, your normal skin. They should only kill the cancer cells. There are several mechanisms in place and several strategies I'm not going to go over here because it makes it too complicated. But this is something where people actively are working on right now to ensure specificity, meaning only the cancer cells get killed. The other thing is, and you've seen it before, that we actually combine the different antigens, the recognition structures on the cancer cells. You know, maybe we make it more specific by taking protein A and protein B, which you can both see on normal cells, but both of them together on the same cell is only for cancer. So that's one way to do it. The other way is we can make it even more effective in many cancer types by combining these super killers with maybe sometimes conventional chemotherapeutic agents, conventional chemotherapy, And then you have your biological immunotherapy with the engineered T cells and the chemotherapy, and can kill the cells. So, this gives you an idea now, not only of where do we need to go and where are we actually actively heading on clinical trial design to really be able to utilize the biological insights and turn them into active drug discovery. But it also shows you where I believe, and many other cancer researchers and doctors, where a lot of the future is heading into the immunotherapy, the immune checkpoint inhibitors, but then more recently even the CAR T cells. So thank you very much for your attention.
Get out from underneath of the thing. Thank you, Dr. Schieke. It's kind of heady, right? Like a lot of science is kind of heady, but it's, I think, so fascinating to see and learn what's really happening kind of behind the scenes that we don't all have access to, and to understand that there's some really great minds behind trying to figure all this stuff out, and we're really making progress. And I think the amazing part is for a rare disease to have this much attention, even though we always can use more, right? And there's always so much work to do, and there's a lot to be done. But for a rare disease to have this kind of work and research being done is pretty amazing, because, if I think of I think the number's now up to 10,000 rare diseases, and 95% of them have no therapies, and they have very, very small patient populations, sometimes a patient of one. So there's no one, and there's no researchers looking into those diseases. So when I hear this, even though I know there's a lot of work, I'm very encouraged that there's actually real work being done to move the field forward. So I think that's pretty exciting. So I think we have, Autumn, we're good. We have a couple of minutes if there are questions for Dr. Schieke before we head to lunch and our networking sessions. If anybody's got a question. Oh, Kelly, okay. No.
If no other hands are going up. So thank you so much for that talk. I love the hope that you give, that not only are we working on it, but we're going to really good places. So thank you for that. As patients, that means a lot, because hope is a powerful thing. I wanted to ask about the CAR T cell information that you were telling us. I imagine that's extremely expensive, but I also wanted to ask, how long would a process like that take for a patient? Because they're working on your own T cells, correct?
Right. This is actually relatively fast. I think you're looking at maybe a couple of weeks or so, because this is every single step that's happening in the lab is what a lot of, basically every single molecular biology lab in the world is doing that. So even though it looks complicated, use a virus, get a piece of DNA into the cell and make them express a new recognizing structure for cancer cells, this is what we do in the lab. We have been doing this for decades. People got Nobel prizes for those techniques many decades ago. So it looks complicated. It is actually relatively straightforward to do from the laboratory side. The price is different, and this is exactly what I was trying to say before, what I did say before, that a lot of the things we can do are basically all on the research side. But we have to make it, but this is a start, right? This is how it is. We figure out with lots of research dollars in the background, what is a new treatment? How can we do things? And then we take it to the regulatory institutions, maybe come up with, over the years, how we can even make that cheaper. Gene sequencing, a whole genome, whole exome sequencing, these things took weeks, I think this was in the 90s or early 2000s. We were so excited that we could do it. It took forever to sequence the genome of one patient. Now we just do it in a heartbeat, and it is so much cheaper. That is something, when I went to medical school, I couldn't do sequencing, in my lab. Now we do sequencing, and a few weeks back, later we have all the data. So, this will all get changed, and with the CAR T cell process, I think you might, in the end, maybe be able to get this done within a few days. Things get much, much faster there. Yeah.
Great. Good question. And we can chat a little bit more about that. I think one of the keys that I'm taking away is how, as the patient organization and the patient community, we can kind of help connect those dots, right, between the research and why it's important, and then how does that really impact us? That's where our voice, and then creating the whole arena around what they call health economics. What's the economic value to a health system in a country if the patients are able to access these new therapies and the quality of life, their activity in the world, their ability to pay taxes, their ability to work? If we can try to connect those dots, we have a better shot at encouraging those who are paying for the therapies to see the ultimate societal value of enabling these therapies to happen. So that's kind of that other piece, right, that has to come into play, because if you have all these great therapies, but no one's willing to make them accessible, or the health systems aren't going to pay for them, then it doesn't do anybody any good, right?
Yeah, no,
So
that's
we've got
a
all those
critical point.
pieces.
And don't expect that from the researchers. We get super excited about the idea, you know, and of course it's very expensive, but that's not our job to figure that out. That is really the society. Ultimately, things will get faster and cheaper, but still a significant cost factor, you know, that has to be taken into account. But so far, we have not seen over the last many years this really slowing down the progress. That's the good thing.
Dr. Schieke, thank you.